This is 9700A2 Biology. Bismillah Rahman Rahim. We start on the topical questions on the A2 chapter of Homeostasis, which is the third chapter in the syllabus. Now let's look at this question. Question number 1. Figure 6.1 is a diagram of a Bowman's capsule of a nephron from a mammalian kidney. On figure 6.1, use label lines and letters to label E, the efferent, G, the glomerulus, and P, the region of the podocyte. Now let's look at a diagram. Now you can see in this diagram, the important thing to remember and revise is this is the afferent arteriole, so this is wider. And this is the efferent arteriole, which is narrower. So you see the lumen is narrower. Now this creates a pressure, and this pressure is going to make the filtrate, which is going to be called the glomerular filtrate. Then I want you to look at the important thing is the glomerular capsule has a parietal epithelium. This parietal epithelium is this outer layer, which is a thickened layer, just like a funnel, supporting it up to here. And then you have the visceral layer, which is made up of the podocytes. And these are the podocytes, which are all, like these hair clips that we women wear. And it is around these blood capillaries. And they're sort of supporting these blood capillaries. So this is something which you must know because it's part of the syllabus. So just pause this video here and have a look at it. Just look at the labels and have a look at this picture, and then we will proceed on to the next part of the question. Now the next part of the question is, of course, this is the labeling which I have done. So you can see E is the efferent, and that's why, because this would be the afferent arteriole. This would be the afferent, A for afferent. But they didn't ask you for that. They asked for E, the efferent, and E is the one which exits. I always say remember it as E is the exit one. So the blood is going to flow out of this, blood is going to flow into here. And this, of course, is a branch of the renal artery. And then we come into the glomerulus, which are the glomerular capillaries. And then, of course, the P is the region of the podocyte cells. Then we come on to describe how the structure of the epithelial cells of the PCT is adapted to carry out selective reabsorption. Now, of course, very simple. Microvilli, this will increase the surface area. Then many mitochondria, because we need a lot of active transport, sodium potassium pumps on the basal side. Then tight junctions, which are the cell membranes where they are sticking to each other so that there is no migration of the channel proteins. Then the folded basal membrane. Why? Because we need so many channel proteins on that side also, the sodium potassium pumps and the facilitated diffusion for glucose to enter the leave the cell and enter the blood and enter the endothelium of the capillary and then enter the blood. Then many transport proteins, co-transporter, many aquaporins because water will follow, and more RER for protein synthesis because they will need a lot of channel proteins, these PCT cells. Now I'm going to show you a diagram on that as well. If you look at this, just a quick revision. Number one, at the basolateral membrane, sodium is pumped into the interstitial space by the sodium potassium ATPase, sodium potassium pump. So 3 sodium out, 2 potassium in. 3 sodium out, 2 potassium in, and then the sodium enters the blood and will be carried away by the blood. Right? Now, so number 2, downhill sodium entry at the apical membrane. Then we have the sodium entering from here into the cell. And then it's again pumped out immediately by the sodium potassium pump. And then, of course, what do we have? We have glucose and amino acids and all this other stuff, entering along with the sodium. And this is called reabsorption of organic nutrients, certain ions, by co-transport at the apical membrane. Apical membrane, or this is the lumen side. This is the basal side. And then 3, reabsorption of water by osmosis through aquaporins. And then we have these aquaporins. So water enters by aquaporins into the cell and then into the blood. And then we have reabsorption of water by osmosis through aquaporins. Water reabsorption increases the concentration of solutes that are left behind. These solutes can then be reabsorbed as they move down their gradients. Then lipid soluble substance diffused by transcellular route. Transcellular route is just the other route which is going to happen as it is because they're lipid soluble substances. So they're going to just go through the cells. Then various ions, chloride, sodium, potassium, potassium and urea diffuses by the paracellular. Of course, these two routes are not in your syllabus in A levels, and you just aware that I was explaining it to you. So transport proteins, ion channels, aquaporins, primary active transport is given in red, secondary active transport has got the dotted line. Secondary active transport means that because of the sodium potassium pump working, sodiums are pumped out, so sodium diffuses it as a secondary to this active transport. Active transport is first, sodium potassium. Then because of that primary active transport, the secondary active transport takes place. So secondary active transport means that the sodium and potassium is going to sodium is going to move in, and that is going to result in the glucose co-transported along with it. So these functions of the PCT are ones which have come in many questions, and I think one just needs to remember them because this is a very direct question. There's no ambiguity about this. A very direct and a very simple question. Then the next one was a fill in the blanks. That was an easy one. Complete the following sentences about osmoregulation. In mammals, the water potential of the blood is constantly monitored by osmoreceptors in the hypothalamus of the brain. When the water potential of the blood decreases, that means the blood becomes concentrated, the production of a hormone called ADH, antidiuretic. Diuresis means a lot of urine. Anti means against it, so less urine. The cells of the hypothalamus increases. So the production of a hormone called ADH in the cells of the hypothalamus increases. The hormone is released into the blood via the posterior pituitary gland. So please understand, this is the brain, this is the hypothalamus, this is the pituitary gland. So it is produced here in the hypothalamus, but then it is released here by the pituitary gland into the blood. I'm just showing it diagrammatically. So this hormone will go into the blood now, but it is released by the posterior pituitary. Posterior means the pituitary gland is a gland like this. It has an anterior part in front. Anterior. You must know these terms. And then there's a back part which is called the posterior, post. These are the different planes we talk about. The anterior means the front and posterior means the back. So posterior and anterior pituitary. Posterior pituitary only makes, the posterior pituitary only makes ADH and oxytocin. Posterior pituitary only makes ADH and oxytocin. That's the only two hormones it makes. The rest of it all like FSH, LH, everything is made by the anterior pituitary. And this causes the kidneys to retain more water until the water potential of the blood is returns to the set point. This is an example of negative feedback. So easy question as far as I can see. Now coming to question number 2, homeostasis in mammals involves negative feedback mechanism. Outline how a negative feedback mechanism works. Now I've always said I've been unable to write this in words, and I usually just would if as an A-level student I would just learn these exact wordings. So first of all, there's a change in a factor away from the normal. The set point or other word which we use is normal. Like for instance, there's a normal blood glucose level, and then there is some change. Either it decreases or it increases. Now this would be detected somewhere in the body by receptors. And this will result in a hormone being released or a nerve impulse being sent. And then this hormone will reach the target organ or the effector. And the effector will then perform some corrective mechanism, some corrective action. And the factor returns to set point. Like giving you an example, you enter this room is very hot. So the senses, the temperature receptors in the skin detect this, and then the blood becomes warm, and then it goes to the brain. Brain sends, okay, let's send impulses to the sweat gland, make sweat. And then let's send impulses to the muscles in the wall of the skin arteries. Vasodilate, more heat lost by radiation. Now you can see in our houses here in Pakistan, we have air conditioners. So there is a sensor in it, detects the temperature. So if you set it at 18, this room becomes 18, well, then it's going to switch off. And then something senses, no, this room is becoming warmer, so it's going to switch on. So negative feedback is like that. Sensors detect, then some corrective mechanism has to take place. and that factor has to normalize. Like your temperature must remain at 37, your blood sugar, blood glucose level must live between 80 to 200. That's it, max. It can't change, so sometimes become 400 and sometimes become 0. If it becomes 0, well, you'll be dead if your blood glucose is 0. Figure 8.1 is a diagram summarizing the role of the nervous and the endocrine systems in the control of blood glucose concentration. So hypothalamus, neurons, alpha and beta cells, pancreas, alpha cells release glucagon alpha, there's an A in it, insulin, beta cells, adrenal glands produce adrenaline. Adrenaline causes an increase in blood glucose. Similarly, glucagon causes an increase in blood glucose, and the insulin results in a decrease in blood glucose. With reference to figure 8.1, when it says with reference to figure 8.1, you must refer to the diagram. You must look at this and read it again and again. Describe the role of the nervous system in the control of blood glucose concentration. Now you look at the mark scheme of this question with reference to figure 8.1. So there are keywords have been underlined without which you wouldn't have got a mark. Hypothalamus detects changes in the blood glucose concentration. Now this was of course given in the question. It said here hypothalamus, so you have to tell me what is the role of the hypothalamus. So hypothalamus detects changes in the blood glucose concentration. This is the first thing that we had to give. Then we said, okay, motor or nerve impulses are generated. Beta cells secrete insulin when blood glucose increases. Alpha cells secrete glucagon when blood glucose decreases. Now, all these underlined words means that they are essential mark scheme points. If you didn't write these words, we are not going to give you marks for these. So, alpha cells secrete glucagon when blood glucose decreases. Adrenal gland secretes adrenaline due to fear. And the nervous control supplements the endocrine control. You know, endocrine is the ductless glands, or the ones which throw their hormones into the blood and then they're carried by the blood to target organs. So, endocrine glands. So this was all part of it, and you can see that this was a 4-mark question, but there are at least 6-mark scheme points. 6-mark scheme points. So any 4, and you would have got the 4 out of 4 if you gave me all these points. So I always tell you to write an extra 1 or 2 things so that if any of your answer is not properly worded, at least you will get the marks for that. Let's go to question part C. If the core temperature of the human body falls, the hypothalamus sends impulses to activate several physiological responses, some of which are listed below. For each one, state how it could help to bring the core temperature back to normal. So if the core temperature of the human body falls, so you fall into a very cold lake and you'd gone boating or something and the boat overturned and you've fallen into cold waters. For each one state how it could help to bring the core temperature back to normal. So the core temperature has fallen because you are in a very cold place or you are in a very cold room. Vasoconstriction and shivering and increasing secretion of adrenaline. How will all these things help? Now there are 3 different headings, but there are 4 marks for it. Now, if you look at the points given here as vasoconstriction, now vasoconstriction is skin arterioles get narrower. Now, the words that I've underlined are the essential mark scheme points. So you had to say skin arterioles. If you said skin arteries, I'm not going to give you any marks for that. So skin arterioles get narrower, so less blood flows through skin capillaries. If you say skin blood vessels, sorry, I'm not going to give you a mark. So less heat lost. So less heat lost because you see, it's just like we want to conserve the heat. You're in a very cold room, you're losing. Your body's at 37, the room is at 10 or even minus 10 or something like that. So you can't afford to lose heat. So how are we going to conserve the heat? So if this room is very cold, you have what options? Okay, let's close all the windows so that you close all the windows, then you turn on the heater. So okay, the heater is the shivering. Muscle contraction. When muscles contract and relax, they release heat or thermal energy, because there's increased rate of respiration. Now then increased secretion of adrenaline. Adrenaline causes increased rate of respiration, aerobic respiration, which is called cellular respiration. So more heat, more ATP generated, more heat released by the process of respiration. So this is what you have to understand is for the three different phenomena: vasoconstriction, shivering, and the increased secretion of adrenaline, which is going to result in, of course, the glucose level will rise because of adrenaline, but that also results in more the rate of respiration increasing, so more energy being released. So this was a 4-mark question, but there were, of course, about 7-mark scheme points. You can see here there was 1, 2, 3, 4, 5, 6, and 7. So you would have to give me one for each heading and then a few extra ones here and there. Coming to the last question, question number 3. The body temperature of human is maintained at its set point of approximately 37 degrees Celsius. If it rises above this temperature, physiological responses begin to return the temperature to its set point. Two of these responses are vasodilation and sweating. Explain how vasodilation and sweating help to return the body temperature to the set point. Vasodilation and sweating. Now, this was the opposite of what we did in the previous question. That was if you're in a very cold place. Now, here it says it is, of course, you're here. How do you return it? It rises above the temperature. So two of these. So vasodilation. So we have to explain that. So in vasodilation, the answer was very simple. Skin arterioles widen, more blood flow to the capillaries. Naturally, arterioles, first artery, arterioles, then it's capillaries, so more blood flow to the capillaries, so more heat lost. In sweating, more sweat will be produced, sweat evaporates, and this, of course, uses heat energy as latent heat of vaporization, and that is going to take away the body heat, and so your body is going to cool down. Then we come to the next part of the question, B part. Diabetes mellitus is a disease where the pancreas is not able to secrete sufficient insulin. The symptoms of diabetes mellitus include a tendency to drink a lot of water and a loss of body mass. Suggest why these symptoms occur. Number one, why do people who are diabetic need to drink a lot of water? And why is there loss of body mass? So let's try to understand this now. Now, the logical answer to the thirsty part is you have to think, why do you get thirsty? You get thirsty if you've had a high protein and a high sugar meal. If you had biryani and you had Pepsi and then you had ice cream as well, well, then of course you're going to really end up pumping the blood with a lot of glucose and amino acids and salts and everything into the blood. So, number one, have high blood glucose concentration in diabetics. That is because their insulin is not being produced, so they have a high blood glucose level at all times. Now, this decreases the water potential of the blood. So I've just made the water potential sign, which is this one, which is the water potential, but you can write it also properly, decrease in the water potential of blood. Now, this will be detected by osmoreceptors of the hypothalamus because the blood is too concentrated. Then this will result in the feeling of thirst. Less glucose is converted to fat because glucose is being lost in the urine. So less glycogen in the liver, less fat being stored under the skin, and the glucose is not taken up by the cells, so the fats and proteins will be respired. Why are we losing weight? That's why people on a keto diet lose weight. So the thing which you've got to understand is, so fats and proteins will be respired. So because you see, people on a keto diet lose weight because they're only eating proteins. They're not eating any carbohydrates. Okay, so high blood glucose, decrease in water potential detected by osmoreceptors of the hypothalamus. Feeling of thirst, less glucose converted to fat, glucose lost in the urine, glucose not taken up by the cells because it's going to be lost in the urine because it's going to filter out, but it's going to not be reabsorbed. The kidney is unable to reabsorb so much of glucose being filtered out. So fats and proteins will be respired. Why you lose weight? Because the fats and proteins are being respired. C part of the question says a person with diabetes mellitus can use a biosensor to measure the concentration of glucose in their blood. You outline how a glucose biosensor works. So you've got dipstick and biosensor in your syllabus. You need to be knowing both of these. Now this biosensor actually has an enzyme called glucose oxidase on these test strips. And then you add a drop of blood to it. And then this enzyme, the blood in the enzyme is on that strip, which you then put in the machine. And the enzyme glucose oxidase reacts with the glucose in your blood, and this is there's a decrease in oxygen because the glucose oxidase catalyzes a reaction between glucose and oxygen, and this drop in oxygen is detected, and this generates an electrical signal, so electrical current generated, and then this is detected by an electrode. So say 100 glucose combined with oxygen, so 100 oxygen drops 200 drops. Now this generates an electric current, and the strength of that electric current is going to result in a digital readout. Now that is the need of the hour these days, biomedical engineering, which is a career which you all should think of, because that is going to be the going forward. We need all these machines, and all these things is the important thing in the next 10 years or 30 years or 20 years. So it gives you a digital readout, a numerical value of the blood glucose. Then the next part, it says suggest an advantage of using a biosensor rather than a dipstick. Dipstick was the one we used it for checking if the urine specimen has a glucose, and it's like a little strip and you put it in it, and then you dispose of that strip because there's a color, there's a chromogen on it, and it changes color. So you can't use that again and again. But this machine, I mean, you need the strips, of course. You have to buy the strips, but the strips, and then you add blood to it, and that strip is, of course, then thrown away. But the machine can be used again and again. So suggest one advantage of using a biosensor rather than a dipstick. Now the marking scheme is very clear. Gives the actual reading of the blood glucose concentration. Actual reading, the accurate reading or the exact reading of the blood glucose at that time. And then, of course, this machine is reusable. You can use it again and again, so the investment is not such a waste. And it's a very precise reading. I mean, it's right to the end, 102 milligram, or 103, or 104, or 104.5, or so more precise readings. Now, there's a difference between actual or the word accurate. Here, the word accurate has also been used. Here the word accurate has also been used. So there's a difference between that and the difference between precise. Precise means to the last bit it is very, very precise. So reusable, more precise readings quantitatively. Completes this topical. I'm going to do another one which is going to have another 3 questions. So this is going to be followed by another second video, and I hope this is helpful to you, for especially the students who are just studied homeostasis from me in school. And I hope this is going to be helping you out in doing these questions and then understanding the syllabus. Thank you very much once again, and thank you for watching.